Hello, my name is Biji Mathew. I am a research associate professor at the University of Illinois at Chicago. First of all, thank you so much for inviting me to give this talk. Special thanks to Dr. Torres and Dr. Salika for extending the invite. Today, I'm going to talk about extracellular vesicles as cell-free neuroprotective agents in retinal neurodegenerative diseases. So I'm going to share my screen. So I'll be, the highlights of my talk is, first I'll be talking about the background a little bit, which led to our current research, and the extracellular vesicles, how they identified their isolation, evaluating their neuroprotective effect, what happens when they are injected into the eye, and then how we can modify or engineer these tiny particles according to our needs. So, as we all are aware, the prevalence of eye diseases in US and worldwide is alarming. Age-related macular degeneration and diabetic retinopathy are two major causes of irreversible vision loss. And glaucoma is the second major cause of vision loss or blindness. Considering the aging population in the world, this number is supposed to get to 200 million by the end of 2050, which is a very serious problem. If you look at the different types of retinal diseases, macular degeneration, as we know, there are two different types of macular degeneration, dry macular degeneration or the wet one. In both the cases, the macula, the center of the retina, deteriorates or degenerates, affecting in blurred central vision and appearance of spot in the center of the visual field. Diabetic retinopathy is due to the hyperglycemic conditions in the eye, which causes damaged or leaky blood vessels, resulting in spots or dark strings floating in your vision, called floaters. This leads to vitreous hemorrhage and ultimately retinal detachment, leading to vision loss. And then there is the glaucoma. The intraocular pressure in the eye increases, damaging the optic nerve and leading to vision loss. So whether it is an AMD or diabetic retinopathy, glaucoma, or any of those conditions, which ultimately leads to a condition called ischemia in the eye. And ischemia generates oxidative stress, resulting in neuronal cell death, neuronal apoptosis, and neuroinflammation. And ultimately, all these different aspects lead to retinal ganglion cell damage. So here in my studies, we are focusing on preserving or protecting retinal ganglion cells or retinal neurons. So the goal, as I mentioned, is to how we can protect or regenerate these neurons. Is it even possible to regenerate neurons? How we can control inflammation, and what can prevent RGCs and axons from the loss. So retina is a very beautifully architectured tissue. It has its layers very systematically and beautifully organized, as you can see here, with the different heterogeneity of its cell types. When a disease condition like here ischemia, that results in displacement of cells in the layers, ultimately leading to thinning of the retina. So the current treatment available so far are intraocular injections, as we know, eye drops or surgery, but they all have its own complications like side effects, high expensive, and can only treat the consequences, not the cause of the pathology. So it is very imperative that we have to develop neuroprotection or the therapeutics which can stimulate axonal growth and regenerate axons, RGCs, and axons, etc. So in 2015, I joined the lab of Dr. Stephen Roth, who is pictured here, and my project was to work with stem cell derived mesenchymal, bone marrow derived mesenchymal stem cells in retinal diseases. So we injected fluorescently labeled stem cells into the retina of rats, normal versus ischemic retina, and followed these animals for nearly a month. And we used optical coherent tomography, OCT, fundus imaging to see where these cells are going. And to our surprise, we noticed that the cells were incorporated over the period of time into the retina. You can see it here in normal and the ischemia, both. We confirmed this result by flat mounting the retinal tissue, and we found that ischemic retina is taking more stem cells compared to the normal eye. These studies are already published a few years ago, and the take-home message from this paper is a single injection after ischemia of these stem cells improved retinal function as measured by ERG, improved histology, and also decreased blood retinal barrier permeability, which can lead to highly increased inflammation. It decreased in neuroinflammation, apoptosis, enhanced the autophagy, and to our surprise, cell remained in the vitreous for a longer period of time, nearly 4 weeks. And also the cells penetrate into the retina, which the penetration was enhanced by ischemia. From here, we know that stem cells are very useful and it has high potential to treat diseases, but at the same time, there are some side effects like limited integration into the tissue. Sometimes it can also cause aberrant growth, forming tumor. They have immune reaction, and there is surgical complications associated. So we then, by then, our lab and other people have already started figuring out and reported that stem cell effects are paracrine, and it is mediated by factors that are released into extracellular space. So towards this goal, we did studies and published in 2 different papers where we have generated conditioned media for using these stem cells. So we did normoxic conditioning of preconditioning of the stem cells and hypoxic preconditioning of the stem cells, and injected both these conditioned media into the rat, into the ischemic eye of the in vivo model, and we found that they have the similar protective effect as the parental stem cells. So this led to the study. By then, it was people have started talking about and reporting that stem cells release these tiny nanoparticles called extracellular vesicles or exosomes into the extracellular space. Then what are these exosomes or extracellular vesicles? They are vesicles, they're secreted by the cells, by all cells, not just the stem cells, all sorts of cells. And they range from 40 nanometer to 1000 nanometer. So based on their shape, size, the smaller particles falling within the range of 40 to 150 nanometer, call them exosomes, and the rest of them are microparticles. So to be safe, in my study, in my talk, I use the term extracellular vesicle instead of exosomes. Both are the same in this case. They are cellular messengers. They aid in communication between the cells, and they transfer materials between the cells. Why are they important? Because they contain lipids, proteins, genetic materials like RNA or even miRNA, and they are from the cells of their origin. And these particles can be engineered. So they can act as cargo delivery agents. They can deliver materials, whatever we want them to carry, and then deliver it into the cells. So that makes them very important. So now you look at the biogenesis of extracellular vesicles. You can see that they are actually formed inside the cell by the invagination of the membrane, and then it, as early endosomes, that takes in materials like genetic materials, cytoplasmic proteins, and surface proteins, etc. And then they fuse together, forming a multivesicular body. And finally, they are released to the extracellular space via the process called exocytosis. The advantages of EVs over stem cells. The most important property is they have the same properties as that of their parental cell or the cells of their origin. If it is coming from a stem cell, they will take on the properties of stem cells. If it is a cancer cell, then they will have the cancer cell properties. They're very, very easy to enter into the cells. They're non-immunogenic. It's very easy to cross-administer between species. They can be easily stored. The contents can be tailored. Their surface can be altered, and they act as small molecule carriers. Based on all these characteristics, they fit for precision medicine very well. So now once we decided to go on the study with the EVs, we figured out how can we isolate them economically and faster. So the gold standard here of isolation of EV is ultracentrifugation. But ultracentrifugation is a little laborious process, and also the yield is not very high. So we developed a method of our own where we combined centrifugation, filtration, and precipitation. So we used this reagent called exosome, which is commercially available, and then coupled with our standardized other methods of filtration and centrifugation. And our yield was very, very good. And also the particles we isolated were, they are definitely having the characteristics of exosomes, which you'll see in a minute here. So this is the characterization of the exosomes. So one method of characterization is the size, analysis of their size. So here we use a nanocyte. Nanocyte is the machine that can tell us the size of this particle. So in this first histogram, which is our method of isolation, you can see that the modal size of the particles here is 8 to 9 nanometer, which confirms majority of our particles fall in the range of exosomes. And there is very few larger particles. Here in the ultracentrifugation method, when we compared, the particle size was a little bigger and not exactly in the exosome category. So our method was fine. Then we also did an electron micrograph study where we can see the shape. They are usually cup-shaped. This is the morphology of the exosomes. We also confirmed using gold nanoparticle labeling, and that also using markers, which also confirms. Here, the another confirmation is using the marker proteins, identifying with the Western blotting. You can see CD63, CD9 are 2 markers. So our particles that we have isolated from this conditioned media fall in the category of exosomes. Now the next question is, how neurons take up these EVs? What is the method of endocytosis of these EVs by the neurons? The first step that we did was to find out whether this process of EV endocytosis is time dependent and saturable. For that, we added, first just to understand the endocytosis, we just added the EVs to the cells and then co-stained with the tubulin. So here another important factor to understand here is these particles can be labeled, fluorescently labeled. So we used a green fluorescent labeling reagent, so they appear as green in our images. So tubulin is red, and you can see how they are co-localized, endocytosis EVs inside the cell. Then we did a time and dose dependent study to understand this endocytosis process, and we concluded that EV endocytosis is time and dose dependent, and it is saturable. At some point, it gets saturated. Definitely, it's a receptor mediated process. We also evaluated the temperature dependency, and we found that when the cells are kept at 4 degrees centigrade, there is reduced EV intake. The next step was to figure out which receptor is involved. Usually, the receptor involved in the process, it can be heparin sulfate proteoglycans or integrins. So the study we did was we blocked first integrins using RGD. It's an integrin binding peptide. So when we added EVs to cells which are already blocked by RGD, we didn't see any difference. The cells were still taking up as much as EVs it could. But when we blocked the heparin, we found that the endocytosis was completely abolished, telling us the endocytosis of EVs by retinal neurons goes through HSPG receptors. So next goal is to find out which microdomain, whether it is caveolin or clathrin, is involved in this process. For that, we just did a simple experiment. Here you can see that we co-labeled the cells treated with EVs with a caveolin antibody. And when we merge the images, we can see that the little yellow parts are the co-localization of caveolin with the EVs. But when we co-labeled with the clathrin, we couldn't see the co-localization or any yellow spots, confirming the clathrin's role in endocytosis. So further confirm this, we blocked caveolin using MBCD. It's a caveolin blocking reagent, and we can see that when we blocked using MBCD, 5 millimolar MBCD, there is no endocytosis. It's completely blocked, confirming this is the final take-home message. EV endocytosis is dose dependent and saturable. It's temperature dependent. It's receptor mediated via HSPG receptors, and it involves caveolar endocytic pathway. So now, since we have identified all these things, we wanted to understand what is the neuroprotective effect of these MSC-EVs in retina. For this, we first used an in vitro system using retinal neuronal cell line. R28 is a mixed retinal cell line, neuronal cell line. So here you can see that all these different markers for retinal cells, like calbindin, GFAP, it's for like astrocytes, bipolar cells, and retinal neurons. So in this experiment, we added EVs, green fluorescently labeled EVs, to the cells, and then we could see that they're all endocytosis. And this just gives you the idea of the R28 cell is a mixed retinal neuronal cell population, like in the retina. It's a mimicking the retina. So our model, the in vitro model, we used OGD, oxygen glucose deprivation. So what we do is like the media media has no glucose, and then the cells are kept in hypoxic conditions, 1% oxygen. So that mimic the ischemia, in vivo ischemia in the cells. So we put them in OGD for 24 hours, then take the cells out and then reoxygenate in the normoxic chamber. And then at the end of that, we will measure endpoints like cell death, proliferation, and we can also collect the cells to do Western blot and everything. So here for treating with the EVs, after 24 hours of OGD, we change the media, add exosomes here, and then put them in normoxic condition for another 18 hours, and then we measure. So we found that first we analyzed the cell death. We found that dose dependently, when we added varying amount of varying quantity of EVs into the cells, it dose dependently protected from cell death, neuronal cell death. Similarly, we did a flow cytometric analysis of cellular proliferation with the cells which is treated, which got OGD and treated with EVs. We found that cell proliferation increased in cells which undergo OGD but treated with EVs. So now we have an idea, base idea of like how that works in vitro system. We wanted to convert it into the in vivo model. So in our lab, we have this in vivo rat model of ischemia where we use Wistar rats. And then at baseline, we take an ERG measurements for retinal function. And then day one, we administer ischemia by raising the intraocular pressure to 130 mercury for an hour, 160 minutes. And then 24 hours later, we inject post-administration, post-ischemia, we inject 4 microliter of this many number of EVs into the eye intravitreally. And depending on what we are measuring, if we have to study apoptosis or inflammatory, neuroinflammatory markers, pro-inflammatory markers, we collect the retina 24 hours post-injection. Otherwise, we'll let them stay for one week. And at the end of the 7th day, we do ERG and then collect the retina for histology and other immunohistochemistry markers. So here is an ERG waveform, which I'm sure you all are very familiar with. So when we flash the eye at varying intensities of light and then record the output from the in the form of, and then it generates the A and the B wave. A wave records the health of the cells in the outer retina, and B wave is the health of the inner retina, and which is coming from this different cell types. It's a biphasic waveform. So here is our data. When we did ERG on our rats, which is, there are 3 different conditions. This is PBS injected ischemic eye, and we also used EV depleted media. That is, when after we isolate the particles from the conditioned media, the leftover media is the EV depleted media, and then the EVs are injected. So you can clearly see that there is significant increase in protection, or there is enhanced functional recovery by injecting EVs into the ischemic retina compared to just ischemia alone. Similarly, with the B wave, we noticed a similar pattern. We also analyzed apoptosis in these eyes. So we did a tunnel study for apoptotic evaluation. This is an ischemia, this is control eye, and this is ischemia alone. And we see that there is increased apoptosis in ischemic eye 24 hours post ischemia and injection of EVs. Here, after we injecting extracellular vesicles, the number of tunnel positive cells significantly reduced. This is, we counted the tunnel positive cells in different layers of the retina, and we found that in all different layers, the number of tunnel positive cells tremendously decreased with injection of EVs into the retina. And this is a data which confirms inflammation is also attenuated by administration of EVs into ischemic retina. So we measured IL-6 and TNF-alpha, which was significantly reduced in EV injected ischemic eyes compared to the ischemia alone. Similarly, we also measured caspase-3 which is a marker of apoptosis. And we found that apoptosis was also attenuated when you inject the eyes, ischemic eyes, with the EVs. So from here, once we understood the protective effect of these EVs clearly, now we wanted to see what happens to these EVs once they are injected into the eye. What is the fate of the exosomes? What particular cell type is taking it in the retina? To do this, as I mentioned, we fluorescently label the EVs with the green fluorescent protein and injected into the eyes of rat, normal versus ischemia, and then followed it for like a month period of time. This is how a control eye, and we used fundus imaging using micron. And day one, you can see a lot of fluorescent EVs are sitting up in the vitreous. And as time progresses, they traverse into the retina. And then even at the end of 4 weeks, you could still see EVs binding to the vitreous. So this led us to the question whether these EVs are binding to the vitreous. Is vitreous acting as a depot for collecting all these EVs? This could be, there can be two effects. One, if it is just binding to the vitreous and just sitting there and not getting released into the retina, that's not good. But at the same time, if they are collected there and then slowly over the course of time, it is released gradually, that is a very good thing. So we wanted to understand that process. For that, what we did is we isolated vitreous from the eye. Vitreous is nothing but it's collagen. And we know that EVs bind to collagen using integrins. So in our study, we isolated the vitreous and then we did a binding assay. And our result here shows that definitely EVs bind to the vitreous with vitreous collagen in a dose-dependent and saturable manner. So next goal was to understand or confirm whether these findings in the in vivo live imaging is translated, or we can get more picture, well identified picture of where these EVs are going. So we did 3D confocal imaging. This is representation of a whole thickness retinal flat mount in the C plane, and we can see that this is day one. The EVs are sitting up in the vitreous, and then as day progresses, by day 7, they traverses down to the retina, and you can see that how they are moving towards the retina. Similarly, we used cryosectioning and immunohistochemistry to understand that same what we have seen before. Here the EVs are green, and these are beta tubulin 3, which stains the retinal ganglion cells. You can see that EVs are co-localized with the EVs are in the layer of retinal ganglion cell layer. And we also wanted to make sure these EVs can reach the optic nerve with our injections. So here, this is day 1. We injected the green EVs, and this is an optic nerve head. And by day 7, we could see some green fluorescently labeled EVs reaching the optic nerve head, which was very promising. We also went ahead and look at different cell types. Which cell type is taking it more, whether they are co-localizing with the retinal ganglion cells. So here, this is a confocal retinal flat mount image, and this IBA1 is a marker for microglia, and green and red, colloquial green is our EVs, and you can see the yellow color, a lot of EVs are taken up by microglia, even in 48 hours. Similarly here, this is vimentin, which is an astrocyte marker, and astrocytes are also taking a lot of EVs, the glial cells. And majorly, we were interested in seeing whether RGCs are being able to take up these EVs. So we used beta-tubulin 3 and BRN3A. It's a RGC nucleus marker, and we looked at ischemic, non-ischemic, and ischemic eye injected. And here in 48 hours, you can see how they are co-localizing, how beautifully these beta-tubulin 3 positive cells have EVs co-localized and giving this yellow shade. And similarly here, the BRN3A positive cells taking up the EVs. And this is a much more close-up of beta-tubulin and BRN3 positive cells taking up EVs. So the summary of this whole study is we concluded that in vitro and in vivo, EVs render neuroprotection by attenuating neuroinflammation and apoptosis. They penetrate through the blood-retinal barrier and reaches the retina and the optic nerve head. They remains in the vitreous for more than 4 weeks, and they bind to the collagenous vitreous via integrins, and they're localized in RGCs, microglia, and other cell types. And this work was published in Biomaterial. This is the paper which explains all these results. So from here, now I mentioned in the beginning that we can generate functionally engineered EVs that, since they can carry small molecules and other materials, we can put our substance of our interest into these tiny particles, and we can send them into the target cell types. I also have mentioned in the beginning that we have hypoxic preconditioned the cells to understand whether the hypoxia preconditioning has more advanced, enhanced effect compared to the normoxic EVs. So here, when we isolated EVs from the hypoxic preconditioned MSC media and evaluated for cell death and cell proliferation using our OGD model of retinal mixed retinal neuronal cells, we found that this is normoxic EVs and this is hypoxic EVs. Definitely, there is an increase in cell proliferation and decrease in cell death with the hypoxic preconditioned EVs compared to normal. This led us to think, what is the material inside the EV which is driving this functionality? We know that EVs contain proteins, lipids, microRNAs, DNA, and all these different things. Out of this, which factor is more important, rendering this functional significance? So there was research already have pointed out towards microRNAs being the entities which is rendering this functionality to EVs. So to confirm that, we used 2 genes, we silenced the 2 genes, which are dicer and agono2. These are 2 genes which are involved in the biogenesis of precursor formation for MIRs. So using an silencing RNA for these 2 genes, we silenced that in mesenchymal stem cells. And after silencing, since I told you earlier, the EVs take upon the properties of their parental cells of origin. So once we silenced this, the EVs isolated from these particular cells, they didn't have any dicer and ago2. So then we use these EVs in our OGD systems to see whether it has the same protective effect as that of the normal EVs. So our result was very interesting. So when we use normoxic preconditioned EVs and hypoxic preconditioned EVs, as I have shown before, the cytotoxicity was decreased because they are normal versus the hypoxic, as we expect. But when we added the dicer and ago2 knockout EVs, the protection was abolished. There was no neuroprotection, confirming the involvement of microRNAs driving the functionality. So this led us to do a RNA sequencing to profile the microRNAs present in these two different types of EVs, the normoxic versus hypoxic. So here is the heat map showing the differential expression of the MIRs. And here is, this is same thing represented as a bar graph. And these are the different significantly regulated, deregulated MIRs. Out of that, we, by using an using MIR walk is a program which helps to identify MIR targets and pathways. So based on the pathways of importance, we came into, we selected nearly few MIRs, but out of that, this particular MIR 424 was very impressive, and it had significant a significant impact in pathways, and this came up as one of the most highly regulated expressed gene between these normoxic and the hypoxic ones. So our next goal was, after understanding this process, we wanted to try to engineer these EVs by overexpressing this particular MIR in our EVs. So we did that by using a lentiviral system. We overexpressed these MIRs in the EVs that we generated from the cells which are transfected with this MIR. And once we had that EVs, we characterized them, compared between the regular EVs with the overexpressed EVs, and we found that the size is no different than there is no change in their size distribution, as well as they express the similar markers, so they are basically the same. And it's been also endocytosis by the neuronal cells the same way as the normal EVs. Here we can see that this is another confirmation. The MIR 424 EVs are dose and time dependently endocytosis, and it reached a saturation. Then we repeated the studies by comparing the normoxic normal EVs versus the engineered EVs. Here you can see that when we use normal EVs in the OGD, definitely there is a significant protection, but that was again significantly reduced. The cell death was reduced when we used the engineered 424 EVs. So, we also evaluated reactive oxygen species to understand how much it can reduce the oxidative stress in the cells. And to our surprise, the FEE 424 overexpressing EVs were much more potent than the normal EVs. And here, these are microglial cells. These are primary retinal microglial cells. And when we studied the nitric oxide production, which is a marker of inflammation, here again the EVs tremendously reduced NO production, ROS, as well as cell death. We also measured inflammatory markers like IL-1 beta, TNF alpha, and IL-6. It's very clear that MIR424 overexpressing EVs are superior in attenuating anti-inflammatory, pro-inflammatory markers induced by OGD. And finally, we injected these EVs along with the normal EVs and hypoxic and the EDM, EV minus condition media minus EVs into the rat, ischemic rat. And you can see here, this is the control, the PBS, and the EDM, which is the condition media minus EV. The green and the red lines are the B wave, which is significantly showing that the functionality is enhanced or improved after we injected these functionally engineered EVs into the retina, confirming they have superior qualities or they have more therapeutic effect than the normal EVs alone. So the take-home summary here is hypoxic preconditioning enhanced neuroprotective effect via differential expression of functionally relevant MIRs. And MSC-EVs can be engineered to package MIRs of our interest. And MIR 424 overexpressing EVs are functionally superior, and hence it is translationally relevant. And functionally engineered EVs can be used in diabetic retinopathy, glaucoma, optic neuritis, or any diseases which involves neuroprotection. And that is the conclusion of my talk. And I acknowledge Dr. Stephen Roth, who is my mentor, and we work together in the lab. And Dr. Sriram Ravindran, he helps with a lot of engineering, bioengineering of these particles, and we have progressed a lot in this direction in different ways, not just functionally engineering the MIRs, but we have studies parallelly going using bioengineering of particles so that we can specifically target to specific cell types, and how to increase the release kinetics, and to get a sustainable slow release into the retina. So those studies are going on. And then thank you to our lab. The people work with us. They're all, and the students, they're all very amazing. They're very hardworking, and the collaborative labs, Ravindran lab. And then we have collaborators from University of Virginia, Shirang. She has the glaucoma model. Shafiq, he's a computational and chemical engineering professor. We do computational biology to understand the localization of this 3D modeling and things. And then Dr. Feinstein, he's a neuroscientist, helps with the, he's an expert in microglial studies and neuroinflammation. And then to the ophthalmology core for helping with the imaging and everything. And thank you to all these people, all the agencies that fund us to keep our research going. And thank you so much for including me in your talk. And if you have any questions, I'm happy to answer.